we'd done this experiment on that had some kind of emotional component to it in this patient. And so that night after the experiment, we were talking and said, there's not much work on emotion going on in neuroscience right now. You should consider that. I said, yeah, I'm going to do that. The light bulb went off and I decided that's what I wanted to do, because that was another professor in graduate school had told several students that were in his seminar, said, well, when you leave graduate school and go out to find your career, do not study the hippocampus, because there's too many people doing that. And do not study the visual system, because there's way too many people doing that, because those were the big hot topics at the time. So that was on my mind. And so when Mike said, there's not much work on in neuroscience on emotions, I'm going for that. So the next step was, how to do that. And there was not really very good techniques at the time for studying, the human brain and psychological functions.
Mm-hmm.
Most of the research involved patients with brain damage of some kind or brain surgery. And you had to kind of wait until you had patients of the right type to ask the question or to answer the question that you wanted to ask. And it could take years to gather the data, in that situation. I was kind of a eager, energetic young man. I didn't have years to wait. I wanted to do it immediately. So I thought, well, there's a lot of thought at this point in time, the early 70s, about emotion systems in the brain. That there'd been some talk in earlier years, but again, there was not a lot of research going on. But there were ideas from the 50s and 40s and so forth. The so-called limbic system theory was very popular and still was at that time. And so the idea was that the human brain evolved these emotional circuits from other mammals, and mammalian ancestors. And so if that was the case, then that would mean that I could study emotional behavior in a rat, and it would be relevant to emotional behavior in a human. I knew there was no confusion in my mind at all. I knew I was not going to be able to study consciousness in rodents, but I did know that I could study the animal's behavior. So I decided to take that task on to go back to what I had done for a few months when I was studying marketing and had done that research that was on rat brains. I don't think I said that earlier. So I went back to rats and started trying to figure out the systems in the brain that would allow the flow of information from a stimulus to go through the brain all the way to the motor system that controls the response. So all the way from the sensory system through the brain to the motor system. Because when you work on split brain patients, you become acutely aware of the flow of information. If you disrupt that flow of information, then everything breaks down. So that was my kind of mental model of how to approach it. Also at the time, researchers like Eric Kandel were doing great things in aplesia, invertebrates, where you had a very simple nervous system and you could trace it from the sensory system all the way through. And it was thought at that time you could not do that in the mammalian brain. It was just too complicated. In fact, Kandel started on the mammalian brain, but says too complicated. And he went to the aplesia, and it took us kind of stuck. It got stuck for a number of years like that. But by the 70s, I was getting started doing this. There were a couple of other labs getting started doing this, different kinds of preparations but it was all using Pavlovian conditioning. And with Pavlovian conditioning, I felt certain that I could follow the stimulus all the way to the response, because it was a very simple stimulus. It was a tone paired with an aversive stimulus. And then the animal, would hear the tone, it would then freeze, and its blood pressure and heart rate would go up. So, okay, it seemed pretty simple. The responses were kind of innate, hardwired responses. That means there would be a circuit that was controlling those. I just had to figure out how a tone comes into the ear and gets to the circuit that controls the behavior. Simple. And it turned out it was actually pretty simple, because at the time, there were new techniques for tracing circuits in the brain. in the old days, you had to like make a lesion in the brain, and then the neurons would degenerate, and you'd have to find the degenerating axons to be able to figure it all out. But with the new techniques, you could inject a tracer in a specific brain area. It would be picked up by the neurons and transported down the axon, and you were able to just march through the brain, making lesions of a brain area, let's say in the sensory system. So I started by lesioning the auditory cortex. The animals could still be conditioned when the auditory cortex was lesioned. All right, so what's going on? We lesioned the auditory thalamus. They could not be conditioned. So at that time, the only connection from the auditory thalamus anywhere was to the auditory cortex. That was what was known. But we said there must be some other connection. And the tracer that we injected into the thalamus like that went to several areas in the subcortical forebrain, the hypothalamus, the caudate putamen, the intralaminar thalamus, and the amygdala. Now, the amygdala had been kicking around in the field for a long time, but never as, there was no understanding of how information would go in or come out of it. But we found this direct pathway from the auditory thalamus, the medial geniculum, into the amygdala. And when we lesioned the amygdala, it disrupted conditioning. And at first, we started with big lesions of the amygdala, but then we said, okay, the tracer is not going to the whole amygdala, it's only going to one part. So we lesioned the part that was receiving the input, and that disrupted everything. So then we injected the tracer to see what that area of the amygdala that receives the input, where it's going to, and it was connecting to other parts of the amygdala. So we lesioned those, and one of them, the central nucleus of the amygdala, disrupted the outputs. And the connections of that to the motor systems was direct. From there, you went to the periaqueductal gray and then down to the somatic motor system. And from the central amygdala to the hypothalamus and brainstem to control blood pressure and heart rate. So it really only took, 5 or 6 years to do all that. Maybe 7 or 8. I mean, it was relatively speaking, to get from the ear to the muscle was, a pretty quick task.